Hi, everyone. My name is Guliz Ozcan, and I'm a postdoc in the real lab. So today I will tell you about how amyloid beta is involved in sleep-wake regulation in zebrafish. Just a little bit of background of Alzheimer's disease. Alzheimer's disease is the most common cause of dementia, and it has symptoms that includes memory loss, difficulties with thinking or problem solving, or language. It's a physical disease that affects the brain, and it's defined by the presence of extracellular amyloid beta plaques and tau tangles. So Alzheimer's disease has several stages. The first stage is the preclinical stage, where amyloid beta levels start to rise. Sorry, okay, so Alzheimer's, the first stage is an increase in the amyloid beta levels, which actually happens decades before the symptoms, any cognitive symptoms, which is followed by tauopathy and neurodegeneration. And this is when clinical symptoms start to appear. So, but at these stages where the diagnosis has been made, there is already major neural loss and circuits have been destroyed. So the window for AD prevention or intervention is the preclinical stages, where amyloid beta maybe has been risen, but we don't see any cell loss yet. Therefore, it's important to focus on these early stages and maybe also understand the in vivo functions of the proteins involved. So there aren't many studies that focused on the in vivo functions of amyloid beta, but recent studies highlight links between sleep and Alzheimer's disease. So Alzheimer's disease patients have overall reduced sleep. They have increased sleep fragmentation, early morning awakenings, increased napping during the day. And also amyloid beta levels oscillate with the sleep-wake cycle. So during the wake, it increases, and at night, after sleep, it dissipates. So it's been shown that sleep affects amyloid beta levels. Sleep deprivation increases amyloid beta levels. But we wanted to see whether amyloid beta levels might also affect sleep states, creating a vicious cycle, contributing to disease pathology. But so here I want to stress, I'm not trying to make a disease model. I'm looking at an acute function of amyloid beta to see whether it affects sleep-wake states. So as Jason introduced, we're using the larval zebrafish, and my methods are basically injecting amyloid beta peptides into the heart of the zebrafish, and then putting them into our behavior tracking system and monitoring their behavior by video monitoring, or also saving some portion of them for looking at their neural activity. So as I said, I'm injecting the heart of the zebrafish, which sounds a little weird, but at this stage, zebrafish hasn't fully formed its blood-brain barrier. Therefore, injected peptides into the heart can go to the brain, which I'm showing here. So this is showing the mean fluorescence where I injected fluorescently tagged amyloid beta, different brain regions, and you can see that it reaches a maximum around 2 hours, and then it gets cleared off. So amyloid beta can form different species. It can exist as a monomer, but it can also oligomerize and form fibrils. And so this is dependent on pH or ionic strength in vivo. And in vitro, if you want to prepare different oligomeric species, if you have colder temperatures and short incubation times, you can enrich shorter oligomeric forms. And if you have higher temperatures or longer incubation times, you can enrich for longer oligomeric species. So basically, this is what I've done. I've prepared 2 different amyloid betas by preparing it at cold temperatures, which forms shorter oligomers as shown by the stem. And if I use higher, like room temperature or higher temperatures, it oligomerizes to form longer structures. And this is quantified here. So then I looked at what happens if I inject these short oligomers or long oligomers into the zebrafish. What happens? So on the top graph, I'm showing the waking activity, and in the bottom graph, I'm showing sleep. And in all my experiments, I've used amyloid beta 42:1, the reverse peptide, as a control. And here in blue, I'm showing the short oligomers effect, which you can see it increases waking activity and decreases sleep. And we see the opposite effect when I use the longer oligomers, where it's more pronounced in the sleep domain. So, and if we quantify this, you can see that compared to the reverse injected fish, the short oligomers cause about a 15% increase in waking activity, and the long oligomers actually cause a 30% increase in sleep. So this is quite interesting, but how does this happen? So for this to happen, the injected peptides must be affecting certain neurons in the brain to cause these behavioral effects. And this is what I'm showing here. Basically, what I've done is, again, inject the peptide and look at what happens to the neural activity of the fish. This is a CFOS staining, and CFOS we use as a correlate for neural activity. Here is the reverse injected controls, and this is when I inject the short oligomers. And particularly, I want to draw attention to this posterior hypothalamus, which is which is one of the wake-promoting centers in the brain. So this was quite interesting because it matches nicely with the wake-promoting effect that we see in behavior. And then if I do the same with the longer oligomers, now we see a general dampening of the overall activity in neurons. And you may be thinking why this looks very different than this. It's because if I just stop the reaction at the same time, all I see is blank brains. So I stain this for like 2 days or so. So, but this is still, this you may think is still a bit artificial because we're injecting the peptides into the fish. So we wanted to see whether if I knock out amyloid beta in the fish, whether I would see some sleep-wake phenotype. And amyloid beta is actually driven from a precursor protein called APP. And when I knock that out in the zebrafish, we see waking activity phenotype both during the day and the night. And again, here I have the waking activity and sleep, this time over 2 days. And you can see the red or the pink here during the day and red at night has an increased activity during the night and a decreased activity during the day. And sleep goes in the reverse direction, although it's not significant. So to conclude, amyloid beta can both decrease and increase sleep. And I have to stress that I'm using low concentrations to mimic physiological levels. And this effect depends on its oligomeric structure. And these different effects on behavior are also matched with differential activity of different neural populations in the brain. And finally, the APPB mutants, which lack amyloid beta, have sleep-wake phenotypes suggesting what I'm observing with my injections may be an endogenous function. Finally, I'd like to thank all the real lab members, specifically Jason and Sumi, our first floor zebrafish labs, and AR UK for funding. Thank you for your attention.
Thank you very much, Guliz, and for sticking to time so well. Question?
Oh, yes. So apologies if I missed this. So what happens if you do sleep deprivation on these zebrafish? Does that increase the amount of these amyloid
So
peptides?
I haven't done those experiments,
Okay.
but based on human and mice, it should.
So this could be like the endogenous sleep somnogen, then, this
Yes.
kind of peptide.
Yes, that's a good suggestion, yes.
Further questions?
Hi, thanks for a great talk. I was just wondering if you knew what the A-beta is actually doing to like electrical activity. So I think there was a paper showing that A-beta regulates orexin circulating levels, but do we know how it's actually affecting the activity of these different neuronal groups?
So we don't know at the moment. I'm trying to dissect the pathway by doing a CRISPR-Cas9 screen. So there are about 20 proteins or so that have been identified as receptors for amyloid beta. So I'm trying to knock those out and see whether I can dissect the pathway, whether amyloid beta signals through one or many of them to cause these effects. But yeah, I don't know on orexin.
I have kind of a technical question. So do you know if the penetration of the two oligomers in the brain is the same in the larvae, or?
Yes, so we haven't measured it, but I think, although it's hard to imagine that the long fibrils could enter the brain as easily as the long ones. So there are two things. One is we see actually a stronger effect with the longer oligomers. And the second thing is we see a stronger effect. And then second thing is there are receptors that carry amyloid beta across in its different oligomeric forms. So I haven't quantified it, whether it reaches at the same level as the short oligomers, but it does reach apparently because we see a behavior effect and also a neural dampening.
Okay.
Hi, thank you for your talk. I was wondering whether you also thought about using a different protein that can oligomerize to differentiate whether the effect you observe is due to some kind of unspecific aggregation of proteins, especially in their oligomeric forms, or to the actual amyloid beta itself.
Yes, so that's a good suggestion. I'm using the reverse peptide as a control. I haven't really checked whether it oligomerizes the same way, but I prepare it the same way. But that's a good suggestion, yes.
Hi, I'm just wondering that in the fish, do you also have 40 and 42 form, and do they aggregate?
So we've tested whether they form by using some antibodies, and they seem to exist, but I cannot really differentiate super well between the oligomeric forms just based on antibody data, because the antibodies can usually recognize a variety of species. So
you haven't tried whether the fish oligomer also do the same thing?
Well, actually, I have tried it in a way by transgenically expressing the fish amyloid beta 42. And it seems to, so I have some variable results with that. That's why I haven't presented it. I'm trying to repeat it. But basically what we see is we are acutely increasing it using heat shock. And then we can see that initially it increases wake, similar to the short oligomeric forms. And then after a few hours, it just reverses the effect. And we see it's an effect that we never saw with any other drug or protein overexpression, so we think it's quite specific. But I haven't taken samples from each set and checked whether they actually oligomerize. The behavioral result suggests it does, but I haven't checked per se.
More questions? Okay, if not, let me just thank again all the speakers for the amazing talks.